Good morning children. In today's class of NS, I am going to continue the question answers that I have started in the previous class. Before entering into the part of the question answers that we cover, we will recall the questions that I have completed so far. So the first different questions that we have covered in this lesson are: what are microorganisms? Microorganisms are the minute microscopic organisms which cannot be seen with our naked eye. So the first question today is about the microorganisms. So they can be observed only through the microscope, that you know very well. So what are the different microorganisms that you have studied? Different microorganisms are bacteria, fungi, algae, protozoa and microarthropods. So these are the different microorganisms that we have studied so far, isn't it? So what are the bacteria? Where do you find them? They are omnipresent. There are different types of bacteria. They are rod-shaped, spherical, cocci, that is so different spiral, so different shapes of bacteria are being present. And next, fungi. Fungi, where do you observe this? The fungi you can observe when you and the moist conditions of the food, or when the food material is under moist conditions, spoils with a black colored spore-like material is formed over it with a black powdery structure, that if you remove it with a needle and observe, keep it on the slide and observe in the microscope, you can see the filament-like structures which is called as, which belongs to fungi. And you can observe the bacteria especially in the buttermilk. So when you take 2 drops of buttermilk and observe under the microscope, you can see the bacillus, lactic acid bacillus or the lactobacillus in the buttermilk. And next, you can also observe these bacteria in the scrapings of your mouth, early morning scrapings of your mouth, and also at the armpits, on your body, so different areas of your skin, etc. You can observe you find the bacteria present. And next, algal varieties. Algae, or mostly they are found in the pond. So algae, you know very well that they are chlamydomonas, spirogyra, spirulina, diatoms. So there are various varieties of algal varieties that you observe, and so these are the algal varieties that you find in the pond. And also you can find protozoans also in the pond. Protozoans like, what are the different protozoans, children? Amoeba, paramecium, euglena, verticella. So these are protozoans. Protozoans like amoeba, paramecium, verticella, euglena. These are the protozoan varieties that you can observe in the ponds. And microarthropods. Microarthropods are the microorganisms. These are also found in the pond water. They are Cyclops, Daphnia, found in the pond water. And you find the scabies mite and eyelash mite also, which causes the skin diseases for human beings. So you find algae, protozoa and microarthropods in the pond. Algal varieties like chlamydomonas, spirogyra and spirulina. So these 3 varieties you find in the pond. Okay, so that's what you have studied in the previous class, isn't it? And what are the different questions? What type of whether the microorganisms are useful or harmful? Yes, microorganisms are both useful and also they are harmful. As we say that they are useful for plants, animals and human beings. They are useful for plants in fixing the nitrogen for them in the soil, and they decompose the material, dead plants and animals, and again return to the soil and act as recyclers, returning the nutrients to the soil. soil back again. So decomposers, they act as recyclers and help the plants to grow well, returning the nutrients to the soil which help the plant. So in this manner we can see that they are useful for the plants. Microorganisms, they are some of the bacteria that is present in the intestine of the animals help for the digestion. So they also these microorganisms, they are also helpful for the digestion of cellulose in plants too. So like this, these microorganisms are useful for plants, animals and also human beings. For human beings, the preparation of food and industry, in the manufacture of different industries also, industrial purpose also, they are being used. If you consider about the preparation of alcohol or wine, the microorganisms is being used. The preparation of food like cakes, bakery products, dosa, idli or jalebi and all. So in the preparation of these products, cheese and all, these microorganisms is being used. Yeast is used as a microorganism, in which we will be studying about this in the next part of the lesson, the story of the microorganisms. And next, so I have studied that they are useful for digestion, they are useful to decompose the dead organisms, they are useful for the preparing the industrial purposes and the preparation of food also they are being used, isn't it? So as much as uses we have studied about the microorganisms, they are also harmful to the plants, animals and human beings too. How they are harmful? We can see that they cause so many diseases. There are useful microorganisms and also harmful microorganisms. Some microorganisms, they cause harmful or dreadful diseases to plants, animals and also human beings. We are going to study about these types of diseases that are caused by the microorganisms in the next part of the lesson. So what are the different diseases that are caused by bacteria, bacterial diseases, fungal diseases and viral diseases and type. So bacteria, diseases caused by the bacteria which are called as bacterial diseases, tuberculosis, typhoid, septicemia, that is lead poisoning, septicemia. Okay, next fungal diseases. Diseases caused to fungal, mainly skin diseases are caused due to fungus. Skin diseases are caused. If you take the plants, smut disease of paddy is a fungal disease that is attached to the paddy. If you take about the red rot of sugar cane and citrus canthus, the bacterial disease that attacks the lemon, and like this there are many diseases that are occurred to the plants also. And next come to the viral diseases that attacks the human beings. Viral diseases are polio, conjunctivitis, that is a disease that is caused to the eyes, conjunctivitis, smallpox, chickenpox. So you know the terms very well, isn't it? Smallpox and chickenpox. And next, swine flu is a disease that is caused due to virus and AIDS. These are the different diseases that are caused due to the bacteria, fungi and virus. Okay. Now the next question really being, what are the human actions which affect the growth of these microorganisms which are useful to us? So what are the human actions in turn? We can see that the application of the pesticides, insecticides and fungicides. Pesticides, insecticides and fungicides, they are applied to the crops where they have the useful bacteria or they affect these organisms, microorganisms. Okay, so excessive use of the pesticides, insecticides and fungicides that are useful for the production of crops will be decreased due to this usage of this. This is the action of human beings which affects the growth of the microorganisms. So that is the answer for this question. Human action causing the death of the useful bacteria and fungi. And next, useful bacteria like nitrogen fixing bacteria that are present in some of the leguminous plants, they behave as a symbiotic relationship between the plants and the bacteria, isn't it? So this bacteria will be affected with the excessive usage of the pesticides, insecticides and so on. And also release of the industrial effluents into the water also kills some of the useful bacteria that are present in the water, pond water or river water and so on. So this industrial effluents should not be released into the, when they are released into the water bodies, they will be affected. So the indiscriminate applying of the pesticides and fertilizers will kill the soil nitrifying bacteria. So on land or in water it will be affected. So human action of indiscriminate uses of pesticides on crops, over usage of artificial fertilizers, that is chemical fertilizers, and releasing industrial effluents after the they affect the death of the useful bacteria and the fungi. So this indiscriminate usage of the industrial, that is chemical fertilizers, they kill the useful bacteria, I said you earlier. So if this continues forever, what happens? It affects the biodiversity. So indiscriminate usage of biopesticides, pesticides are chemicals that are used, insecticides and fungicides. They kill the nitrogen fixing bacteria in the soil. Nitrogen fixing bacteria present in the soil. Okay, children? Here the industrial effluents, release of industrial effluents, wastage. Industrial effluents means industrial waste either into the soil or into the water, pollutes soil and water, and kills the bacteria and fungi that are present in the soil and water, which is useful for decomposing the material, decomposing of organic matter. It affects the decomposing of organic matter because it kills the bacteria and fungi that are present in water and soil. So it will be affected. So if the decomposition, the decomposers are being affected, so due to the industrial effluents, then it leads to the that is human act. Due to these human actions, it leads to the imbalance in the nature itself. The ecological balance will be hindered, and also it leads to the imbalance in the biodiversity also. Okay, so this is the reason where the human actions affect the growth or leads to the death of the microorganisms which are useful to plants, animals and also human beings. So this is the answer for that question. Hope you have understood this, children. And next question is, why the cooked food spoils soon but not uncooked food? Uncooked food or raw food. In uncooked food or raw food, the action of the biochemical reactions of the metabolites present inside the enzymes will be inactivated, but during the cooking process the enzymes activation takes place, the biochemical metabolites of the enzymes takes place, action takes place, and so it decays slowly. So in the cooked food when compared to the raw food. Okay, so that's why cooked food spoils soon when compared to the uncooked food. So next, what questions would you ask to know about the bacteria? So bacteria, what are the different types of bacteria? Where are they present? Can we see them? What is the most common shape that we see? How many shapes of bacteria are there? Is there any relation between the shape and the disease that is caused by the bacteria? And is the bacteria with no specific shape? Like that you can ask different questions for that. Next, what will happen if you add buttermilk to chilled milk? Chilled milk means cool, very cold milk. If you add the buttermilk to cold milk, then milk does not convert to curd. So when milk converts to curd, in generally, when it is warm milk only converts to curd. And what develops over the growth of lactic acid bacteria converts the milk into curd. Lactic acid bacteria converts the milk into curd. This does not form in the cold milk. If you add buttermilk to cold milk, it does not form. This does not form. Okay, so that is the case over there. Next, visit any milk bakery or milk chilling center and write about the techniques that they use. Milk chilling centers or the bakery. Today's questions, children. In today's questions, I am going to share you about the different techniques that are undergone in the milk chilling centers. In the milk chilling centers, the processes like pasteurization, curdling of milk, ripening of milk takes place. Here, in the pasteurization technique, the harmful microorganisms are killed. And in the curdling and ripening of the milk, the useful bacteria are used for conversion of the milk into curd or milk into cheese. Okay, so this is how the techniques are used by using the microorganisms. So here the microorganisms are killed. Microbes are killed by heating technique. They heat up to 72 degrees centigrade and suddenly they cool it and they bring bring back the room temperature and distribute them and in the packets for distribution. Okay, so this is how the pasteurization process takes place in the milk chilling centers to preserve the milk for the longer period. They use this technique in the milk chilling centers. Next, curdling of milk. Curdling of milk, they use buttermilk to convert the milk into curd. So here lactic acid bacteria is used to convert the milk into curd. Okay, so useful bacteria acts over it to convert the milk into curd. Curdling process takes place. Okay children, just a minute. So here in curdling, boiled milk is curdled. That is, you use warm milk to curdle it. Milk is stored by adding the bacteria. In the ripening of the milk protein is added. Some microorganism is added to the milk protein that is, to convert it into the semi-solid substance called cheese. So here cheese is prepared by ripening of the milk by action of the microorganisms. So these are the techniques used in the milk chilling centers where the microbes are killed as in the pasteurization technique to store the milk or for the longer period. And here these two techniques, curdling of milk and ripening of milk, are done to prepare the curd and the cheese and so on. So these are the different techniques used in the milk chilling centers. And next, next question is about the permanent slides that are of the microorganisms to be seen when you return to the school. Children, you will be shown these slides. Okay. Next, prepare a model of any microorganism and write a note on them. So you can write any microorganism. For that, you can do TMV, that is tobacco mosaic virus. You can do a model, or you can also prepare a model of chlamydomonas. If you want to do the model of chlamydomonas, children, you can take a cardboard of oval shape. You can cut the cardboard into oval shape and draw it over it like this. And outer side of the oval shape, you color it with the paint, green color. You color with the green color, children. Color with the green color. And take a round button of red color over here in the center here, and two blue colored buttons you take and stick over here, and two wires you can take and stick at the top of the oval shape. Correct. Okay, over the cardboard. This vise acts as flagella. This vise acts as flagella, and this oval shapes body structure. The center, the red colored button acts as nucleus. These are contractile vacuoles. Two blue colored buttons, and here you can see the chloroplast, which is green color. Okay, so like this you can prepare a model of chlamydomonas, which is belongs to algae. And next I told you we can also prepare the model of that is virus, that is TMV, tobacco mosaic virus. You can see this picture carefully, children. So this is the bottle you can take for TMV, tobacco mosaic virus. Tobacco mosaic virus. For this, you can take a bottle where turn the page number, children, to find out the virus. Just a minute. Okay, see here, this is tobacco mosaic virus. For this you can take a bottle and take a pieces of paper cut in oval in shape like this and stick to the bottle or a glass jar, any bottle you can take and stick it as shown in the figure, and then take a wire or a spring you can take and connect it at the top, which acts as RNA, and these paper strips which are cut in the oval shape as protein components. Okay, so for this you can prepare a tobacco mosaic virus model also with this. As you know very well, viruses are the interlink between the living and non-living organisms. They are living inside the body of the living organism and non-living outside the body of the living organisms. And they cause various diseases. They can be seen only with the help of electron microscope only. Diseases like, as I said earlier, polio, conjunctivitis, swine flu, smallpox, chickenpox, AIDS, and all. These are the diseases that are caused by tobacco mosaic virus. You can also prepare the model of HIV also with a ball. Paper balls you can take, a rubber ball or a paper ball you can take and stick on the paper balls to the rubber ball and prepare like this model of HIV, human immunodeficient immunovirus. Okay, electron microscope of view, how you see under the electron microscope, this model. This also you can do it as a model children. Next, why should we clean our hands with soap before eating? Why do you clean our hands with soap before eating? Because microorganisms are present everywhere. They are omnipresent. They are present on our body, in our hands, etc. When we eat, so we will be working with different things. And so we have to clean our hands before eating. Otherwise, the microbes that are present in our hands enter our body through the food that we eat when we eat the food without washing our hands. So it is necessary to maintain the healthy hygienic practices to avoid the microorganism enter our body. So we clean our hands with soap before we eat anything. Any particle, anything, if you take in, before eating anything, you will be washing your hands perfectly with soap and then proceed to eat. That is the case. And several other questions that are asked in this lesson are as the preparation of the models, glass slide models to observe the bacteria. Bacterial observation I said, that is lactic acid bacteria. For that, we will be taking observation of the lactic acid bacteria. We will be taking a glass slide. During the first step, you will be adding the 2 drops of buttermilk over it and cover that. Add crystal wine. First, 2 drops over the glass slide. Second, you add that is heat it so that it doesn't move. Spread it with spatula and heat it in the second case. Then add 2 drops of buttermilk in the first section. Then heat it, spread it and heat it. Then in the third case add a methyl orange or any other color indicators, methyl orange or crystal violet, to observe the cells clearly or the organisms clearly, and cover it with the cover slip. And if you want, you can wipe out the things that are present outside, and while putting the cover slip, make sure that there are no air bubbles form inside. So cover it with the cover slip so that the organism doesn't move. Okay, and then observe under the microscope. This is how you will be doing, preparing the slide for any observations. In the similar pattern, for observing the algae, you will take the green scrap, greenish, move towards the greenish pond and take the pond water and observe the organisms present in that water under the microscope. You can be saying the algal varieties like, as I said, Chlamydomonas, Spirogyra, Spirulina, etc. So Spirogyra will be like this, they will be spiral shaped, and Spirogyra will be in this shape. So observation of these slides is very much important. Okay children? You have to observe it carefully. These are Spirogyra. Try to draw the pictures neatly in your notes. Okay, Spirulina, Spirogyra. Okay, try to draw the pictures and be prepared to identify the pictures whenever you get it in the examination. Next question is here about the protozoa. How do you observe the protozoa in the laboratory? So then what you have to do, you have to take a hay, you have to soak them in the water and prepare a decoction of it. Then the protozoa, the development of protozoan takes place after a few days. Then you take that water and observe under the microscope. So this is how you can observe the amoeba, paramecium or euglena, anything like structures under the microscope. Protozoans, which are amoeba, you know it. Amoeba is irregular in shape. It contains the nucleus, vacuoles and cytoplasm in the center. It is a fluid, liquid fluid that is present inside the body. Okay children, this is how you have to know about the different microorganisms that are present in our daily life. Okay, and next, coming to the next question is about the, the rod-shaped structure, that is lactic acid bacteria, that how you observe under the microscope. If it is lactic acid bacteria, what is the structure of the lactobacillus? Rod-shaped, isn't it? So this is the structure of the bacteria that you observe under the microscope. So if you see, it will be like this. Rod-shaped bacillus structures. So this is lactobacillus that is observed under the microscope. Its shape it is rod-shaped. Okay. Next. So how do you find the bacteria? I said in the buttermilk, in the mouth, in the early morning scrapings of the mouth, and under the armpits. And also you can see over our skin also, you can see this bacteria under the bark of the tree, and also you can observe the bacteria. And what is an experiment of observing the microarthropods? For observing the microarthropods, soil is collected from the field and add water to it in a beaker of glass and stir it continuously and allow the soil to settle. Afterwards, take these 2 drops of water and put it on the glass slide and observe in the microscope. Then you can see this mites, different insects that are observed under the microscope. So this is about the different questions that arise in this lesson, children, and hope you have known it. And in this lesson you learnt about the Antony van Leeuwenhoek. Antony van Leeuwenhoek was a fabric merchant, and the biology, the microbiology, the story, the invention of the subject relates to his invention of microscope itself. As a science was, microbiology as a science was born in the year 1674 when Antony van Leeuwenhoek observed the first microscope which was used by him was a single length microscope. Okay, so that you have to write about it. Who invented microscope? Antony van Leeuwenhoek. What were the major groups of microorganisms? Bacteria, fungi, protozoans, algae, etc. These are the major groups of the microorganisms. Where do viruses reproduce? Viruses reproduce only in the living cells, that is host. That hosts living cells only. Diseases caused by bacteria and protozoan diseases, you know it. Like protozoa, malaria is caused by protozoa, amoebiasis is caused by the protozoa. Malaria and amoebiasis is caused due to the protozoa. Next, when do viruses behave like living organisms? They behave like living organisms in the body of the living organism, host cells. Who called bacteria as animalcules? Antony van Leeuwenhoek called the bacteria as Leeuwenhoeks. Name the microorganism present in the buttermilk. What is the microorganism? Lactobacillus is the microorganism present in the buttermilk. And next, coming to the next question is, write about the diseases that you know it. Chicken pox or any other diseases that are caused by the microorganisms. You can write fungal diseases, allergies, ringworms, etc. that are caused by fungus. Skin diseases, as I told you. Protozoan diseases, malaria, amoebiasis, and microarthropods causes scabies, I said you. Viral diseases, bacterial diseases, I said so many times, you know it very well. So our body is a good example for symbiotic relation with microbes. So there are several bacteria, isn't it? How can you say that our body is a good example for the living of microorganisms? Means many of the disease causing ones live in symbiotic relations with other bacteria. There are different kinds of bacteria in our intestine which are useful for our digestion, and some bacteria help in the synthesis of the vitamins too in our body. So that we can say that our body is also a place for the microorganisms. So you know very well and understood this lesson, and the next lesson will continue about the story of microorganisms, part 2 about the vaccines and discovery of the vaccines, antibiotics and the scientists. Okay children,